Hello, this is Danny Pineda. I'm the medical director of oncology and Invitae. I was part of the ACMG ClinGen copy number variation interpretation work group during the development of the guidelines. Have been part of the ClinGen group since the beginning, actually before the beginning, when it was the ISCA consortium. And what I want to discuss today is how at Invitae we had implemented the guidelines for interpretation of intragenic CNVs. As a disclosure, I am an employee and stakeholder of Invitae Corporation. And to go next and to tell you a little bit, give you sort of context of what Invitae is, and so we can discuss how we have implemented the guidelines. I'll tell you a little bit about Invitae. So Invitae is a large diagnostic lab that really does testing, genetic testing across many clinical areas, including fertility and prenatal testing, pediatric testing, neonatal testing, adult genetics, including cancer, cardiology, neurology, proactive testing and screening. We also have, as part of that, cytogenetics tests that include chromosomal microarray, chromosomal analysis, FISH. But the vast majority of the tests that we do are panels, are next generation sequencing panels, and they are all diagnostic grade and they're used in diagnostic and proactive settings. And we also have a medical exam. So just to focus on the intragenic CNVs, I'll be talking about the panels and how we do NGS panels. So we start like any other lab with DNA extraction, then we target the genes, and that targeting is unique to us and it's optimized for our needs. Then we sequence, and then it's pushed to a bioinformatics pipeline to extract the variants, and then those variants are classified through a rubric, and we'll talk a little bit more, that is based on the ACMG guidelines, called Sherlock, and the paper is cited there. One thing that is important is within the design of our NGS panels, we thought it was very important to have a single platform to detect both sequence variants and copy number variants and from the single NGS run. And the way we do this is analyzing the read depth of every single exon across all the genes that we target on our assays. And what we do is basically fix a baseline and see how that baseline deviates from the majority of the patients in any given patient in any given exon. So if a patient has more reads than the baseline, that would implicate duplication or infer duplication. If the patient in the opposite would have less reads for that given exon as compared to the majority of the patients, that there we could infer a deletion of that exon or several exons. And that's the way that we determine copy number variants from the NGS data in general terms. Next, I want to show what the talk is about. Here, what we would need to do is to apply the rubric that was put forward by the ClinGen and ACMG guidelines group. And here are sort of the CNVs that we want to cover here. Those are the ones that involve the 5' and 3' end or one end of the gene. The ones that are within the reading frame of the gene for both deletions and duplications. First step to do that is to determine the effect of that CNV in the reading frame of the gene. We had a extensive webinar series, and I went extensively on how to do that in that webinar. So I would encourage you to go through that link and review that, and that will give you elements on how to do that. But in general terms, for genes that cause haploinsufficiency, disease due to haploinsufficiency, what you want to demonstrate is whether the variant leads to a nonsense-mediated decay or not. Some deletions would result in a frame shift and lead to nonsense-mediated decay and then lead to haploinsufficiency, and some will be in frame and not lead to nonsense-mediated decay, and the gene may actually work normally if the delete region is not, or the disrupted region is not key for the gene's function. And during to calculate the impact and adapted to a rubric, we took the sequence variant interpretation group rubric that modified the PVS1 criteria for CNVs, and we adopted that. And it essentially, as I explained, we want to know if a deletion disrupts an exon or multiple exons, and those are biologically relevant for the gene and cause nonsense-mediated decay. They will get the most weight and will be classified as like pathogenic by themselves. Or if there's a deletion that is multi-gene deletion, but either the effective reading frame is not disrupted, meaning that it's in frame, so it's going to take a chunk of the gene, but it's not going to lead to an absent allele. But then you have to demonstrate whether that piece of the gene that is deleted is essential or not for the gene's function. If it's essential, then you would apply more points and make it probably like pathogenic. If it's not essential, or if it's not known to be essential, then you'll be skewing that to less points and probably classify that variant as variant of uncertain significance. But let's say that there are a deletion and it's in frame and it affects a portion of a gene where there are actually other transcripts that have that exon absent. That probably is not going to lead to disease. That probably is not going to disrupt that gene, and it's probably not going to get any points, and the variant would be classified as a variant of uncertain significance, and it's probably going to be benign at some point once you have more clinical data to support. For duplications, and here I'm going to stop a little bit, it is important to note that since these are duplications that occur within a gene, are very small, they could be as small as a few hundred base pairs, up to a few kbs. The mechanism for these duplications to happen, in the majority of them are going to occur in tandem. So even if we cannot prove that the duplications or the gains are actually in tandem, it is safe to assume that most of them will be in tandem. And if you assume that they're in tandem, then you can classify the variant that way, assuming that the variant will be in tandem. Very few of them will not be in tandem inserted somewhere else, and we accounted for that in our rubric and have a little bit less weight than for deletions. So let's go through it. If the variant is proven in tandem, that's going to occur very rarely, only when you're really doing whole genome sequencing that you can actually characterize where the breakpoints are and where the insert is going to be. Then you can apply the highest score. But if the variant which we apply to the majority of the tests is going to be presumed in tandem, and again, you have to know where the duplication starts and where it stops, and you need to make sure that that's occurring within the gene, then you can say, yeah, it is disrupting the reading frame and it's going to lead to nonsense-mediated decay, and you can apply strong. Depending on the gene's mechanism, you can make that variant up to likely pathogenic, applying 0.9 points, and that's the case in most of the genes that cause that for those duplications that would lead to nonsense-mediated decay, and are in genes that cause disease due to haploinsufficiency. But then if there's no impact on the reading frame, you shouldn't apply points because you're just inserting something more, and that may not disrupt the protein at the end. It could, but it's not necessarily true, and those variants should be classified as variant of uncertain significance by default. So how have we implemented this in Vitae? So as I mentioned at the beginning, we created a system called Sherlock. And really what Sherlock is it took the sequencing guidelines from 2015 and it refined the guidelines. So the sequencing guidelines were sort of general, they didn't have many rules. So what it did is took all those 28 criteria that were put forth by those guidelines and refined them into hundreds of different criteria that are very detailed, and they have a very detailed use case for every single of them and have very, very specific rules. So what that allows is consistency in the classification of variants between different variant scientists. And even if this were to be applied broadly to other laboratories, you would expect that people who would use this rubric across different laboratories would classify the variants in the same exact way, and the variants would be classified consistently. The system was initially vetted in 500 genes and over 40,000 variants. We now have observed way more than that and have way more than those genes in our menus where we use Sherlock. Importantly, the system is being refined as time goes by, as we learn more about biology of different genes. So the system is semi-quantitative and it uses a scale of points similar to the CNV guidelines, except that it's integers. And it moves from pathogenic points when you add evidence that proves pathogenicity, and benign points for evidence that proves that the variant may not cause disease. You need to have 3 benign points to make the variant likely benign and 5 benign points to make it benign. You need 4 pathogenic points to make the variant likely pathogenic, or 5 pathogenic points to make the variant pathogenic. And we use the main 5 levels of evidence. That's if the variant is present on the population databases, including NOMAD, the type of variant. So here we'll be looking at CNVs, but these are just examples of how we look at it. Clinical findings, so if we have seen it in unrelated cases with the same phenotype, or segregating in families, or if we have seen a variant, a variant in trans with pathogenic variant in a recessive gene, etc. And then we use molecular studies, both experimental and in silico, to predict whether the gene's function is disrupted, and we give different weights to it to arrive to a final classification. Here's more or less the interpretation rubric for CNVs, and it's very similar, as you can see, to the modified rubric that was put by the sequence variant interpretation work group, except that this was done way in advance from when that rubric was published recently in 2018. So let's go through a couple of examples here. So here we have a duplication in the BRCA1 gene. As you know, this gene is associated with breast and ovarian cancer, also with prostate cancer and pancreatic cancer. And what we have here is a duplication of exons 17 through 19, and this would lead to an out-of-frame duplication that would result in nonsense-mediated decay. So let's see how we classify this with the ACMG guidelines. So we know that the guidelines allowed for a range between 0.4 and 0.9 points, and since we know BRCA1 is a gene that causes disease due to haploinsufficiency, and that it disrupts the reading frame and result in nonsense-mediated decay, and there are similar duplications that would cause disease, and we have clinical support for that, we can make that variant likely pathogenic. And let's see how that compares to Sherlock rubric. So Sherlock rubric, we have that same duplication. So that duplication in our rubric, we assume that is in tandem and that is rare, and we get 4 pathogenic points. And the reason is because it will be disrupting the reading frame and leading to nonsense-mediated decay, and that would lead to a likely pathogenic classification. So as you can see, it is consistent between the two rubric. I would like, because there were questions about terminal gene duplications and how we view those, I want to just give an example on how we view those. So for terminal 5' or 3' duplications, what can happen and often happens is that if you have that tandem duplication, so let's say exons 1 and 2, and then you put it next to it, you would expect that that reading frame gets reconstituted. Therefore, those variants in general are thought to be variants of uncertain significance because they are not expected to disrupt the reading frame of the gene and can be reconstituted. Because they reconstitute the reading frame. So I'll give you a tricky example here. And bear with me because it could be difficult. So here we have a 5' terminal duplication of the APC gene. So it goes from promoter 1A all the way through exon 7 of the gene, actually part of exon 8. And here we had actually discrete breakpoints, and we knew that that it was in tandem actually. So it went from promoter 1A all the way to exon 8, and we know that it stopped in exon 8 and then reconstituted in tandem. So this is a view of what happened. So this is the duplicated part. So as you can see, it starts from exon 1A and then breaks here, and then then you have the intergenic region that inserts in exon 8, and then it starts again promoter 1A, 2, 3, 4, and then reconstitutes here. So that, if you look at that, up to there, you have a reconstituted gene, and you should not expect this to cause disease. So how would you classify that? Duplication includes the initiator colon, and you apply 0.45 points, and that would give you a variant of uncertain significance. In Sherlock, same. It includes the feathers exon. You would expect that it would reconstitute the reading frame. It gives you some weight, but not a lot, and classifies the variant as a variant of uncertain significance. But lo and behold, we found a paper. And what we found in this paper is that if you put distance between promoter 1B and the start site of the gene, promoter 1B is it's stronger than the 1A. And primarily the transcription of the gene comes from promoter 1B. So if you put distance between promoter 1B and the start site of the gene, that gene doesn't get transcribed. That the gene that is reconstituted is not getting transcribed as efficiently as it could be. So what happens is the first part of the duplication actually gets translated more efficiently. If you can look at here, what happens is that the transcription will start here and then get to exon 8, and then it will read through, and that will lead to a frame shift and nonsense-mediated decay instead of causing instead of going through here and transcribing the reconstituted reading frame. So it can lead to this. So taking that into account, that what is being actually transcribed is a product that has a frame shift, then you can apply 0.9 points and make it like pathogenic. And there actually was clinical information on those patients who carry different types of these 5' duplications that evidently had familiar adenomatous polyposis. So it allowed to classify this variant as slightly pathogenic. And same with the Sherlock rubric. Now you have a duplication that disrupts a critical region because you're putting that distance between the promoter 1B and the start of the reconstituted reading frame. And that also allowed a classification of likely pathogenic. And with that, I'm going to end. I hope this brings a little bit more clarity on how we do interpretation of intragenic variants. This one is difficult, so we'll keep going at it. I want to thank the work group and my colleagues for putting this together. I would like to thank the Invite team, both in the variant interpretation and the oncology team, for supporting this. Thank you.